The following is part 2 of an excerpt from a Reddit post made by an individual claiming to have the full truth on what we call alien greys. In this online interaction, they even answered questions from people in the comments section. A link to the original post can be found in the description of this video. Make of this information what you will. Speaking of genetic engineering, following sequencing of their genomes, we noticed a troubling and universal characteristic in the 5' of the regulatory sequence of each gene, which we call the Tri-Palindromic Region. The TPR are 134 base pair sequences containing, as its name suggests, 3 palindromes. In genetics, a palindrome is a DNA sequence that, when read in the same direction, gives the same sequence on both DNA strands. They serve both as a flag and as a binding site for proteins. The 3 palindromes in the TPR are distinct from one another and have been poetically named 5' TPR, mTPR, and 3' TPR. The TPR is composed, in 5' to 3' order, of 5' TPR, 12 base pair spacer, chromosomal address, 12 base pair spacer, mTPR, 12 base pair spacer, gene address, 12 base pair spacer, and 3' TPR. The chromosomal address is composed of 4 base pairs and is identical in each TPR of the same chromosome, but distinct between each of the 16 chromosomes of the genome. The gene address is a 64 base pair sequence that is unique for each gene in the whole genome. It's therefore understandable that the TPR serves as a unique address not only for numerically identifying a gene, but also for identifying its chromosomal location. For those with only a basic knowledge of genetics, this is completely unheard of. No living thing in our biosphere has this kind of precise address in its genome. Once again, the presence of TPR cannot be explained by evolutionary pressure, but only by genetic engineering on a genomic scale. TPR opens the door to several possibilities. One of them suggests that ebiogeneticists can insert or remove a gene from a cell in a way that is far more targeted and efficient than our technology allows. No proteins have been identified in the ebio genome that interacts with TPR. Rather, we believe that these proteins are exclusively targeted by external genetic engineering tools, probably used at the zygotic stage of embryonic development. The nature of these tools is unclear, but we definitely don't have anything like them. The probable absence of these proteins from the genome is a further indication of their artificiality. Given the high probability of artificiality of their genome and the apparent ease of modifying it with biomolecular tools, it's not out of the question that there could be polymorphism between individuals depending on their role and function. In other words, an individual could be genetically designed to have characteristics that give it an advantage in performing a given task, like soldier ants and worker ants in an anthill. Note that these previous statements are speculation. To my knowledge, only one individual genome has been sequenced. I can't make a definitive statement on genetic variation between individuals. I've talked a lot about intergenic regions. Now I'll briefly discuss intragenic sequences. Briefly, because there's not a lot less to say, despite its obvious importance. Much like ours, their genes have silencers, enhancers, promoters, 5' UTRs, exons, introns, 3' UTRs, etc. There are many genes analogous to ours, which is not surprising given the compatibility of our cellular machinery. What's disturbing is that some genes correspond directly, nucleotide by nucleotide, with known human genes, or even some animal genes. For these genes, there doesn't seem to be any artificial refinement, but rather a crude copying and pasting. Why they do it is nebulous and still subject to conjecture. There are also many genes which are not found in our biosphere whose role has not been identified. Finding the purpose of these novel genes is one of the aims of the program. I'd like to note before going any further that this heterogeneity of genes of known and unknown origin is an undeniable proof of the artificiality of EBOs. To conclude with genetics, the mitochondrial genome, at the time I was working there, had not yet been sequenced. It's safe to assume that this genome would also be streamlined and possibly has some version of TPR, transcription and translation, and protein expression. I briefly introduced the differences in post-translational modifications between human and EBO. This is hardly a surprise, as we often see the same thing between different terrestrial species. Obtaining a viable protein from a DNA sequence is a complex process involving hundreds of protein intermediates, each with a precise and essential role. A minor variation in this assembly line can lead to functional irregularities in the final product. So, it's no surprise that there are setbacks along the way when the first EBO gene transfection attempts failed to produce the desired functional protein in human cell lines. Fortunately for us, the work of what I imagine to be another team at another site has led to the development of an EBO cell line named EPI-G11, derived from epithelial tissues. With this tool in our hands, we were able to transfect and overexpress proteins of interest in order to eventually purify and study them. For your information, we use a biological ballistics delivery system, also known as a gene gun, for our transfection needs because other methods are not very effective with cells of this line. For example, the viral vectors tested cannot be internalized by EPI-G11, and lipofection is too lethal. EPI-G11, like most eukaryotic cell lines, enters a phase of exponential growth when exposed to fetal bovine serum. It's only half surprising that a cell line from such an exotic source should be sensitive to the growth factors present in FBS. In my opinion, this can be explained by the addition of animal genes to the genome, such as growth receptors. Gross anatomy. They are morphologically very similar to the gray aliens that are part of modern folklore. Their height is about 150 centimeters. They have 2 arms, 2 legs, and a head. Still, there are some notable differences. Skin. The gray skin that is often described in folklore is in fact a biosynthetic film, which likely serves to protect the EBO from a hostile environment. It doesn't provide effective protection against temperature changes, but it does offer adequate protection against the passage of liquids. It's possible that this film confers other advantages, but my knowledge on the subject is limited. Under the gray film, the epidermis is rather white, and the texture is very regular and without any hair. We do not see any defect other than the folds near the joints. It's described as greasy in one report, but that's not something I've observed. The same report states that a strong, lingering smell of burnt hair and ammonia is present when the film is removed. There are a lot of pores on the skin, crossing from the epidermis to a gland in the hypodermis. These glands and pores are the terminal part of the excretory sudoriferous system, which could explain the previously mentioned smell. Head. The head contains 2 large oversized eyes, 2 nostrils without protuberance, a narrow mouth without lips, and 2 ear canals without auricles. There is a mandible, but the musculature is vestigial. There are no teeth or tongue in the oral cavity. The nasal cavity, where the nostrils meet, is compact and does not rise cranially, but extends axially. There appears to be no equivalent to the olfactory bulb in the nasal cavity. The mouth leads directly to the esophagus, and the nasal cavity to the trachea. The trachea and esophagus do not communicate. Eye. Like the skin, the eyes are covered with a semi-transparent biosynthetic film that offers the same environmental protection, while providing protection against certain wavelengths and light intensity. When the film is removed, a more traditional eye is revealed. It's about 3 times larger than a human eye, and there are no eyelids. The size of their eyes suggests they have excellent night vision. It seems paradoxical to cover them with a semi-opaque film. Perhaps they only need to wear it in a bright environment. Their sclera is the same color as their skin. The iris is pale gray, and the pupil is black and oversized. The lens is rounder than a human, and the musculature used to adjust focus is more developed. On the retina, there are at least 6 types of cone cells. The responsiveness of each of these 6 types of cone is specific to a wavelength band, with a minimum of overlap between each other. The result is a broader visible spectrum. Ear. As mentioned, the outer ear has no auricle, and the ear canal is unremarkable. The inner ear has all the characteristics of a typical vestibular and cochlear system, although the curvature of the cochlea is more pronounced than a human. This probably results in greater hearing acuity for low frequencies. The brain is tetraspheric, that is, composed of 4 major sections. The sections are separated by transverse and longitudinal fissures and are connected to the central lobe, which acts as brainstem and cerebellum. The volume of the brain is around 20% superior to that of a man of the same height. It has a much more pronounced level of gyrication than an average human. Moreover, the ratio of glial cells to neurons is also slightly higher than in humans. It is important to mention the presence of nodules on the central lobe. Histological analysis of these structures reveals a kind of intricate biological circuitry. It is speculated that these nodules are essential to interact with their technology. Consequently, determining the proteome of these structures is an absolute priority for the program. The neck is proportionally longer than that of a human, and at the same time, relatively thin. As mentioned, the esophagus and trachea are separate. There are no vocal cords in this region. The musculature of the thorax is underdeveloped. Muscles equivalent to the pectoralis major can be seen. We can also see the trapezius and deltoid muscles. The sternocleidomastoids are well defined. The ribs and sternum are clearly visible. There are no nipples. The abdomen is wider than the thorax and bulges slightly forward. There is no navel. The pelvic bones are apparent. There are no genitals or anus. Their hands have 4 digits, including an opposable thumb on the medial side. They have no nails, and the texture of their fingerprints is composed of concentric circles. Fingers are proportionally much longer than in humans. Unlike humans, finger musculature is entirely intrinsic to the hand. In other words, the muscles used to move the fingers are not in the forearms, but entirely located in the hands. At first glance, the feet consist of just 2 digits, but a necropsy soon determined that each toe was made of 2 fused digits. The medial toe is marginally longer than the distal toe. The feet are relatively longer and narrower than in a human. Their musculature, however, is vestigial. This concludes the reading of this excerpt portion from the Reddit post. What did you think of this story? Do you think it's real? Leave your comments below and make sure to like and subscribe to UFO Zone for a notification when Part 3 comes out.